I'm Stuart Wolfert, Director of UCLA Conversations. I've really been looking forward to tonight's program since October, when our distinguished speakers agreed and accepted our invitation to speak with us tonight. Dr. Stephen Clarke is a UCLA Distinguished Professor of Chemistry and Biochemistry, and he's former Director of the UCLA Molecular Biology Institute. Dr. Clarke has been honored by the American Chemical Society, the National Institutes of Health, the American Society for Biochemistry and Molecular Biology, and UCLA's Academic Senate, which has awarded him the Eby Award for the art of teaching. Dr. Clarke's laboratory studies biochemical mechanisms that recognize molecules damaged in the body for their removal before they cause disease. Dr. David Eisenberg is UCLA's Paul D. Boyer Professor of Molecular Biology, a former director of the UCLA-DOE Institute for Genomics and Proteomics, and a Howard Hughes Medical Institute investigator. Dr. Eisenberg is a member of both the National Academies of Sciences and Medicine. He's among the world's most highly cited scientists. Dr. Eisenberg studies the molecular basis of Alzheimer's and Parkinson's diseases and therapeutic approaches to potentially limit their pathologies. Please join me in a warm welcome for Professors Stephen Clarke and David Eisenberg.
Well, it's wonderful to be here tonight, and I feel especially humbled in the presence of Professor Fernando Torres-Gil, one of the world's leading experts in aging. So, talking about the molecular basis of aging, I first want to give a big shout out to Stuart Wolfert. Stuart and I have been colleagues and friends for most of my time at UCLA. He's done a fantastic job of bringing the contribution of scientists at UCLA to the public, and I've enjoyed working with him. So a big shout out to Stuart. So I've been at UCLA since 1978. I used to tell my students that I was teaching here when your parents were in diapers. I'm now afraid that's going to be, I was teaching here when your grandparents were in diapers. I hope you don't get that wrong. And I'm very happy tonight to have some of my relatives here. My wife, Professor Kathy Clarke. My sister, Lorraine Young. And my brother-in-law, Stephen Young. So, and I also want to shout out to the wonderful collaborators I've had at UCLA. Where's Harvey? Here's Harvey Hirschman. Harvey another pioneers in the field of protein arginine methylation in cancer. And then the work I'll describe today, I have some wonderful collaborators, including Stephen Young, a couple of young whippersnappers in the department, Jose Rodriguez and Joseph Liu, as well as David Eisenberg. So what I'd like to do tonight is tell you how one biochemist looks at the human aging process, and others would tell very different stories. And so I like the story of the Indian parable of the 7 blind scholars and the elephant. And so the 7 blind scholars are looking at an elephant for the first time, and one of them has his hands around the legs and says, "My colleague's an elephant, it's just like a tree." And another colleague who is the trunk says, "I'm sorry my distinguished colleague, but you're quite wrong, an elephant is like a hose." Okay, so you get the picture. And so if we look then at the aging elephant, we have some people looking at aging as stem cell biology, or telomere biology, or cellular senescent biology. But here I am at the right, looking at aging as molecular damage. And I think the picture, it's all right, okay? All of this is a part of the molecular basis of aging, and I think one needs to know it all. But what I'm going to tell you about is the molecular part. Okay, so here's a spoiler alert. Okay, this one slide is going to be everything I'm going to tell you tonight. Okay, so this is the deal. Biomolecules that make us up are inherently unstable. And they're falling apart, and as they fall apart, they're not functioning any longer, which leads to them to change in physiology. Okay. But, here's the big but. Organisms have evolved to recognize the kinds of damaged molecules and can, in many cases, limit the accumulation of the damage and the functional losses. So, bad news, good news. So, if one looks at the molecules that make up life, beautiful molecules of DNA, beautiful molecules of RNA, beautiful molecules of proteins, and I think, David, are you going to show us some films of molecules and the beauty of them?
I'm going to show some really bad molecules.
So anyway, so these are the molecules. They're biosynthesized in perfect form, but as soon as they're made, they're starting to be degraded. And over time, you have degraded forms of these molecules that are less functional, and basically you're going towards, sort of back to the atmosphere. And I think I show here, simple molecules like glucose with 6 carbons. Here's glucose, and you can imagine chemical bonds, it's a little bit of vibration, and once in a while they break, okay? And when they break, that molecule then is no longer. So here is DNA with, much larger, 1 billion carbons in a chromosome. Here's a protein with tens of thousands of molecules of carbons, oxygen, and they're all sort of leading to these very simple molecules of carbon dioxide and water. So we're sort of back to basics, okay? These molecules, all life came from thin air. Plants and bacteria were able to take carbon dioxide and water and nitrogen from the atmosphere and make us, okay? But we're going right back there with the damage. So that's the bad news. We're being betrayed by our own molecules, okay? And that's leading to, global changes in physiological functions. And I'm not sure how many of you can actually see this laundry list. This is a list of things that go wrong with aging. And every year I check off a few more for myself. And so the news is that physiological functions peak at about the age of 18. Okay? Unless you're Stanley Corman, and then, So I love telling my students that, they'll look at my gray hair and I'll tell them, well, listen, you're on the downhill already. So anyway, the chemical question that we ask often is we take chemicals, we mix them up, and we incubate them at some temperature, and we see what happens. So we do chemical reactions. So this is the chemical reaction that 16-year-old freshman from college, and put in the incubator at 37 degrees for 59 years. And so my question is, what the hell happened? And so the point is that the molecules aren't stable. You get your drugs, you get your food, they have expiration dates. And those expiration dates aren't 100 years. Those expiration dates are 6 months or, a year or so. And, pity the poor undergraduate who gets a shipment of a research protein and leaves on the bench overnight, comes to dry ice, leaves on the bench overnight. You've ruined it. That protein sat, for 12 hours at room temperature. But we're expected to sit for, for 100 years at 37 degrees. So, one thing that is clear to me is that it's not all bad. And I love this slide of, this is an older slide of record marathon times versus age. And what you'll see is that, people at like 30 years old actually have better record marathon times than people that are, 18. So, okay, you hear? All right, okay. So, and in looking at this, you can go and you can look at toward the end of it, and there's someone my age who is running a marathon with 6.5 minute miles. Okay, so doing well. Okay. Okay, all right. So now here is Muscle Beach, 1949, the year of my birth. And I know you're all looking at that beautiful creature in the air, but I don't want you looking at her. I want you looking at the old geezer below her, okay? And so that old geezer, he has a suit on. He was probably in the air just a little bit earlier, but he's just not photogenic, all right? So we actually do well. And one of the things that people have thought is that, in fact, there's an evolutionary pressure for us to keep youthful in aging. And it's what people have called the grandmother effect in wisdom. So, basically, after we're done having children, what's our role? Our role is protecting the tribe and taking care of the grandchildren and making sure that there are good decisions being made. And so, don't go to war with that tribe, that's not going to work out well. Don't eat that plant. And so that's the wisdom. And so my argument, and this is controversial, I think I'm not going to get much grief from this audience, is that there is a wisdom in aging, and there's an evolutionary pressure then to keep us young. Okay, so molecules degrading, an active fight to actually prevent that. And so the degradation is not inevitable, but it's a loss mitigated to a greater or lesser extent by countermeasures. Okay, and one of these countermeasures is well appreciated. Our cells have actually have two molecules of DNA, one from mom, one from dad. If anything happens to them, and probably in the time that we've been sitting here, we've all accumulated a few mutations in our DNA. But we have of the 20,000 genes in humans, about 400 of those encode proteins that look for damage. And they look for damage on the DNA. They go up and down the DNA strand and look for damage. And when they find damage, they repair it. Okay, so you can keep then the DNA in a good form. What's not so well appreciated is that proteins also are subject to damage and small molecules. So DNA may be the blueprints, but the proteins are what's doing things that are functional in cells. So the proteins are long strings of 20 amino acids. So here's one protein that's hopefully working for all of you right now. Trypsin. So the trypsin in your small intestine hopefully is this protein that's now digesting the protein content of your dinner, and it does so with long strings of 20 amino acids. Which have different structures. Now, what's good for a protein is not to be in contact with oxygen. Oxygen is horrible. Oxygen is reactive, degrades. Glucose is horrible, degrades. And water degrades. So all these things that we absolutely need are actually leading to degradation. And basically, it's really time itself that's the problem. And so, of the 20 amino acids, there are two bad characters. 2 amino acids are put into the genetic code, but that was a big evolutionary mistake. And the reason it was a mistake is that these 2 amino acids, aspartic acid and asparagine, have reactive side chains, and they can react with the polypeptide chain itself and lead to a whole variety of chemical damage. And so I showed here in the green the structures of aspartic acid and asparagine, and then the arrows are chemical reactions that happen not in 100 years, but happen in days and hours. And so making derivatives that are deamidated, racemized, isomerized, and kinked. Okay, so that's the problem. And in fact, David and I collaborated on a recent paper where we looked at actually at what happens with peptides that can form amyloid that have isoaspartyl residues, which have the kinks. And what they are is that they actually form amyloid much faster, and they form these wonderful structures that David pioneered where molecules come together and they can exclude water by just coming together absolutely perfectly and making then bonds so tight they don't come apart. And so are you going to tell us more about that, David?
Yes.
Okay, good. Okay, so that's just my introduction to David. Okay, so that's all bad news. How does the organism defend itself then? And one of the ways it defends itself with one of my favorite small molecules, S-adenosylmethionine. S-adenosylmethionine I love because you can actually buy it at Costco. And you can chew on it, and it does everything good. It's actually a pretty good antidepressant. Ameliorates the pain of arthritis, and actually, if you're abusing your liver in any way, it can actually slow that down. So what does S-adenosylmethionine do? Lots of things, but one thing it does is transfer methyl groups, the small CH3 group, to a whole variety of acceptors, and one of those acceptors are damaged proteins. And so here are 3 of my early graduate students in a reflective mood, and what they found was that this reaction didn't occur with normal proteins, it was selected for damaged proteins. So we found an enzyme that can actually recognize damage. And so here is a representation of that molecule that Professor Todd Yates determined, and this molecule turns out to methylate the damage and lead to its conversion into normal forms. It's a repair enzyme. So not only DNA gets repaired, but proteins get repaired. So one of the things that we want to ask is, we've discovered protein repair, this is great. How can we figure out what it does? So we enlisted the help of Professor Stephen Young, then at the UC San Francisco, and made mice that lacked it. And so the mice actually normally live about 2 years. The mice were all dead after about 42 days. And I said, Steve, this is fantastic, okay? What happened, okay? Did they turn gray? Did they start walking funny? Did they start talking stupid to their children? What were the aging phenotypes of these mice? And it turned out it was seizures, and it turned out that, it was a complicated story. We could actually have ways around the seizures, and these mice lived. And that's one of the toughest things for a biochemist, because you discover something you think is the secret of life, and then you take it away, and life goes on. So that was sort of hard to mull over, but it turns out that if you can't do repair, organisms don't take it on the chin. They have backup pathways. And in 2022, we discovered this backup pathway, and the backup pathway is, if you can't repair it, degrade it and make a new one. Okay? You repair it first because that's easy. If you can't do that, you degrade it. And so we have a class of ubiquitin ligases that this one that we discovered would specifically look for damaged proteins that weren't repaired and then take them to the amino acids and take it again. And so we have redundancy in aging, controlling the accumulation of these guys. And so the conclusion is, aging organisms are tough. They're going to fight back against chemical damage. And I love this example. Some of you may have heard it, trying to figure out how something works. So trying to figure out how an organism works, and taking something simple. And so you're given a tool, and your tool is a pair of wire cutters. And you want to know how a toaster works, okay? So you take the wire cutters and cut the cord, and you plug it in, and what happens? Nothing, okay? So you know that, you need electricity to make the toaster work. Now you take something more complicated. You take the Mars lander, okay? And you take your pair of wire cutters, and you clip one random wire. What happens? Nothing! Yes, that's nothing, because engineers have made it like life. They've triple engineered it, so if one system fails, another takes over. And I think that's the lesson that we're learning in aging systems. In fact, the evolutionary pressure is enough so that things actually keep going. Okay, so bottom line is that we take care of damage. Now, let me just end. One of the things our fantasies are, can we do something? Now we know about these processes, can we make it better somehow? Can we sort of increase the activity of these repair enzymes? Okay, and the answer is that we don't know anything. But, at meetings of aging, someone comes up with, these are the things you need to do to live a long time. And it gives a whole bunch of chemicals. And at the end of the meeting, then, sometimes it's myself, sometimes somebody else stands up and says, listen, how is this any better than listening to mom? Okay, so mom, good diet, exercise, and sleep. So, how much of that might work through molecular repair? And, what can we do about it? We can do things about these things. And in fact, diet, I think we had a pretty good diet tonight, but one of the things in diet, lots is controversial. One thing that's not controversial, fruits and vegetables are magic, okay? We don't know what that magic is, but, okay, there's something magic. Okay, and then you can go down the list of, coffee and vitamin D and omega-3 unsaturated fatty acids. Probably good, there's some controversy. And then you go down the list to, alcohol and ascorbic acid, vitamin E, more controversy. But anyway, the idea is that, you might be able to do something to keep these enzymes going. And so at the end of all of it is, I'm on the left-hand side of this fountain of youth. And what I'm hoping is, there's something in that broth, okay, that might help get to the other side and come out on that side. So, thank you.
So Steve, what's your advice about nutraceuticals? You mentioned S-adenosine. Should we rush down to Costco?
Yes. Okay. You know what? It depends what you've got, okay? It depends what you have. So I think, for people that have depression, they'll go to a psychiatrist, and they'll tell a psychiatrist, I want to take a drug, and the psychiatrist says, well, why don't you take S-adenosine, which actually works almost as well. It costs about 100 times more, but, that works. Now, that other list I had, I think, well, actually, I am amiss at not mentioning coenzyme Q. Okay, I wanna not sleep on the couch tonight. So Kathy works on coenzyme Q, and I take that religiously. Okay, along with vitamin D and S-adenosine. So
you can get that at Trader Joe's.
I must say that I had problems walking for a long time with my bursitis, my greater trochanter, and so I started taking S-adenosine, and it went away. I also started doing yoga. So, but the thing is, I have enough faith in S-adenosine that I don't want to do the experiment of stopping taking it because I don't want to start hurting again. But that's if I was a real scientist, I would do.
We'll give you a placebo.
You need David, do you want to come over here?
No, I'm happy. Does this mic work? You can hear me?
Yes.
So you'll, I think, see slides. You have it upside down. That's that all. Now, can you see slide?
Yes.
Yeah. Well, I thought Steve's talk was rather optimistic. Forget that for my talk. So I'm going to be talking about neurodegeneration, why it happens, and why we don't yet have drugs for it, but maybe why we can get drugs for it. I'm going to start with little personal stuff about UCLA. I second what Steve said about the wonderful opportunities here. He mentioned collaborators. I mean, it's really great to be in a place where we have our fundamental science right next to the medical school, right next to the School of Engineering, where we meet faculty and students from these various disciplines, and very easy to establish collaborations. Steve mentioned some of his collaborators, and here's a list of some of mine. And I also want to say how important it is that we have this wonderfully diverse and interesting group of students who enliven the lab every day. So this list of people from our lab come from 7 countries, and every day at coffee at 3:30 we have discussions not only about science but everything, and it's just extremely stimulating. UCLA is a great place to work. So I want to say a little bit about how I got into this area. My dad was a pediatrician in our little village, and for him, relieving suffering was the main reason for working, for being alive and helping. And he intended that I'd follow him into medicine, and he had followed his uncles. But when I got to college and had a tutor, he said, if you want to relieve suffering, then study biochemistry. You can do much more than being a physician, where you'll see 10, 100, 1,000 patients, but you could affect the world if you're a scientist. Well, I believed it, my father didn't. And so I went into science and came to UCLA. And I have to say, I spent my first 30 years here. By my standards, Steve's a baby. And I was enjoying working learning the structure of proteins and other biochemical molecules, along with Jim Lake, who's here, and other colleagues. Bioinformatics came along. It was such a revolution. We could make a discovery every day when the human genome was sequenced. It was a great time, and I was really enjoying it. But then, about 30 years ago, in the late 1990s, suddenly it dawned on me, I certainly hadn't done what my tutor in biochemistry had said, and what my father had wanted me to do. I was having a great time, but maybe I should think about how I could do something that would affect a greater portion of the population. So I looked around for diseases that maybe I could contribute to. And here were a whole group of diseases, namely those that all were connected with fibers and neurodegeneration. Alzheimer's disease, 7 million patients in the United States. Parkinson's, nearly 1 million patients. FTD, we don't hear so much about, but another one of these deadly dementias that strikes younger people, and of course ALS, and another 45 or 50 diseases, all associated with the same sort of fibers of different proteins. Each disease has its own particular protein fiber. At that time, 30 years ago, there were no effective drugs for any of these diseases. Today, there are no effective drugs for any of these diseases. But what has changed is we start to have atomic structures for them, and that's a good step towards drugs. Now, I was careful to say that these diseases are associated, each one with a fiber. There was a very smart guy 400 years ago named John Locke. Maybe you've heard of John Locke. And Locke argued persuasively that you can never tell the difference between association and causation. So do the fibers really cause the disease, or are they just associated with it? But I think the case for causation with these fiber diseases is quite strong. So in the case of Alzheimer's, the protein is called tau. In the case of Parkinson's, it's called alpha-synuclein. Each one forms fibers in the brain in those diseases. And here's the evidence. There are very rare families who have a mutant form of the protein tau. And they get Alzheimer's disease at very young age, like 40s, some even in 30s. If that mutant form of tau is put into mice, now they get a Alzheimer's-like disease. Well, we can't test them, their cognition very easily, but they mess up their cage and behave badly. The same with alpha-synuclein and Parkinson's disease. Those, if you give them the mutant form of alpha-synuclein, which causes severe Parkinsonism in humans, you give it to mice and they get movement disorders. So I think that's a very strong argument for causation, and I'm going to show you two others. So in the upper left, you can see a picture of some of these fibers. In this case, those are Alzheimer's fibers. And experiments done at UCSF in the upper right, where they have for patients look for the amount of tau fibers in these patients who have been diagnosed as Alzheimer's patients. It says tau PET, it's a measure of the fibers in their brains on the horizontal axis. I don't know if you can see my cursor, but on the horizontal axis, tau PET, on the vertical axis, brain atrophy. And you can see there's a correlation, that is, the patients who have a lot of fibers in their brain is atrophied, and they are demented. Even in individual patients, you see that. So that says to me, if we could stop the formation of fibers, maybe we could halt, or maybe we even reverse Alzheimer's disease. And one other thing that says the same thing to me is that we are now able to get magnified pictures of these fibers. So the fibers in the upper left, those are magnified about 10,000 times in the electron microscope. In the lower right, it's more like several 100,000 times magnified. We're looking at the course of the tau protein. We're looking down the fiber. We're looking at one slice through the fiber. And every Alzheimer's brain that we've studied, and now it's dozens, if we extract the fibers from the autopsy brain that some family has donated to the brain bank, we see the same fiber, always. If you have Alzheimer's disease, you have this fiber with this structure in your brain. So again, that says to me, if we can stop this fiber from forming, we can maybe halt the disease. So that's what we're up to. So how do we do that? Well, here there's more pictures of Alzheimer's fibers. And what we found is that there is a small molecule. This is, it's called EGCG. It's in green tea. And that molecule has been known for a long time, found by others. It will break up these fibers. And if we look at this column here, this fibers from the brain at the top, after 3 hours, most are gone, and the ones that are left don't look in such good shape. After 24 hours, we only found one fiber of many, many hundreds. So EGCG really breaks up the fibers, but it's been tested many times as a drug. It's failed every time it's been through human trials, and the reason it fails is it's very water-like. You can see OH around it. Water is H2O, so it's water-like. It can't get into the brain. Our brains are fatty. They're covered with lipid, and EGCG doesn't get in the brain, and so it's not useless. It's useless as a drug. But Paul Cited, postdoc, he had a good idea. He said, let's trap the EGCG working on the fibers after just 3 hours. So we'll see how it works. We'll find out how it works, and now maybe we can then get other molecules which are more drug-like if we see how EGCG works. And what he found is that EGCG binds to these tau fibers, forms a column in this crevice between the two different columns of fibers. So here we're looking down a fiber with 10 or 15 layers, but in our brains, the fibers will have thousands, tens of thousands of layers. And that's where the EGCG binds. So what good is that? Well, now we know what the binding site is. And we call it a pharmacophore. Pharmacophore means it's a place where drugs might bind. So now we ask, are there other molecules that might bind there that are more brain-friendly? So Kevin Murray, who was an MD PhD student in our lab, he's now a neurologist at Brown University. He took 61,700 molecules, which we thought to be brain-friendly, that could enter the brain, and he puts each one in this pharmacophore space, and he sees how well it binds. And these are the molecules that, at least theoretically, by the computer, bind well here. EGCG is what we call a positive control. It binds well, and so do these others. He orders the ones that bind well. We go to the catalog. He found some 47 of them, and we test them. And sure enough, some of them, like CNS11, so-called, yet when we put it on the Alzheimer's fibers, it breaks them up. So now we've got something that's brain-friendly and seems to break up the fibers. It does break up the fibers. Another one that came out was CNS11G, similar but different, and it breaks up alpha-synuclein fibers, the Parkinson's fibers. Here are the Parkinson's fibers taken from the brain of the Parkinson's autopsied patient, and CNS11G breaks those up. And here's a worm. You can get worms that get a Parkinson's-like disease, and when they do, they get these fluorescent dots in them. We feed the worm CNS11, and most of those fluorescent dots disappear, meaning the fibers are gone. And CNS11G, all but one of the fluorescent dots goes. So now we have small molecules, and these molecules we consider small, or molecular weights are such that they can be formulated as pills. In principle, we can make a pill. What is pharma doing about Alzheimer's disease? They make antibodies. Why do they make antibodies? Well, that's what they can make. So they have a hammer, and they use that hammer no matter what the nail is. They say we're going to cure Alzheimer's with antibodies. It doesn't work. Antibodies get into the brain very sparingly. They don't get into cells. And you have to give them as infusions. Patients to take the Lilly drug, you have to go to the hospital once every 2 months. How are we going to treat 7 million people? Sending to the hospital every 2 months? No, impossible. We need a small molecule that can be formulated as a pill. So I'm almost done here. I'll just tell you that we've learned about the mechanism. That's the work of a graduate student, Jiawei Liu. And here's the Parkinson's protein. Again, we're looking down the fiber of many thousands of layers. This is the Parkinson's protein alpha-synuclein. And here it is bound to CNS11G, the molecule that breaks it up. And that CNS11G, it forms a fiber that it runs right along the fiber of the Parkinson's protein. And that fiber, the two fibers together, they're fighting one another. And when the small molecule fiber gets long enough, that's when it snaps the Parkinson's fiber. So I'm going to show you, this is my last slide. This is now using another form of microscopy called atomic force microscopy. And on the right side, you see an atomic force microscope, a schematic of it. You have a cantilever, and it has a very fine tip. You buy these tips, they're made of silicon nitride, and they get down at such a fine tip, it's as small as molecules. And now that cantilever, it bangs up and down, bing, 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 and it runs across whatever your sample is. The sample actually moves underneath the tip, and then there's a laser that reflects off a mirror on top of the cantilever, and now you can see how high the object is on the cantilever. And the higher it is, the brighter it is. And here we've got a Parkinson's fiber, and we're starting to measure it after we've added our small molecule. And we follow it for about 12 minutes as it's coming apart. And that's going to be a movie that you're going to see in the central part. Ooh, I hope you're going to see here. Let's see. Yeah, there it is. So we're watching it over 12 minutes compressed. You can see that this small molecule takes apart the fiber into pieces, and now those pieces are taken into smaller pieces, and then the smaller pieces into still smaller pieces, and so forth, and that fiber is completely degraded. So what I've tried to show you is that through these structural methods, we've identified small molecules, meaning molecular weights below about 400, and short peptids. I didn't go into that, both of which take apart fibers and detoxify them from both Alzheimer's and Parkinson's proteins. The mechanism I didn't go into except to say that both of these form the small molecule and peptids. They form fibers which then attack the pathogenic fibers. In the background of all these slides, you can see some of these Alzheimer's fibers. I didn't mention that they are exceedingly stable. They're stable in boiling detergent. So it's pretty amazing that these small molecules and small peptids can take them apart. But they can. And they can because they form fibers. So what's the step from these molecules to a drug? Unfortunately, there's still a lot of steps. So in our case, we're collaborating with UCLA chemist, Patrick Heron. He's made variants of the molecules that we have. We're testing them to get ones that are more brain penetrant and have good efficacy and are also safe. And we've had tests in mice, which are encouraging. Working with mice for us is very expensive because you need a lot of mice to get a good answer. So we frequently run out of money. We're hoping now to work with human cells instead of mice. And then if we get to that stage, which might take about 3 years, we might be in position then to apply to the FDA to do a phase 1 test, would be a test on humans to see if whatever molecule we have at that time is safe. So there's still lots of steps. Of course, right now, as you've heard, future funding is very uncertain, so we may not be able to do it. I just wanted to show you again the list of people who are in the lab, and here's some of their faces. Paul Seidler, who now he's a professor at our neighboring university, USC. David Boyer is an expert in electron microscopy. Jiawei Liu discovered these fibers of small molecules. Well, I guess we're over here. Kevin Murray is now a neurologist. He says he can do a phase 1 for us if we get there. Michael Sawaya is an expert in computational work. And Lisa Lutter did the atomic force microscopy where you can visualize breaking up of the fibers. We've had funding from NIH. It's been very strong. Now suddenly it's completely uncertain. We put it in a huge request to do the work that I'm telling you about, and we were told that that grant won't even be reviewed. They won't even look at it. That's the situation. We have other funding from Spark NS. We've had wonderful support from our Department of Pathology, who supplies the brain tissues. And of course, we depend on donor families. I'm very grateful to them. So I probably told you a little bit more about this than you wanted to hear, but I hope it's an interesting story.
Well, thank you very much, Professor Stephen Clarke, Professor David Eisenberg. So let's take some of your questions now. Who'd like to start? Okay, let's start over here. I'll give the microphone.
Hello. Thank you. My name is Edward Keenan. I'm a linguist here. I have a totally naive question. I understand, perhaps wrongly, that some animals live much longer than others, like lobsters. Would it be true that they have more stable biomolecules or fewer tau fibers?
Actually, I know in the case of whales, they do have systems for getting rid of these fibers, and some whales, I think, live like 200 years. So at least some species have been able to evolve systems that are helpful. And Ed is a friend, neighbor, expert in linguistics.
Okay.
How long are the peptides that interact in these proteins?
7 resin.
Right. So very specific.
Why is it?
Wasn't it a young woman in your life?
Because that particular sequence of 7 residues forms fibers that then bind to tau fibers. The monomers bind and then form fibers against the tau fibers in a metastable way, creating a strain which then breaks the tau fibers.
And I'm Professor Emeritus of Biostatistics. Is it possible that maybe some drug company can help you with the clinical trial that you have in mind?
That's certainly a hope. I've tried to get support from drug companies. You may know that both Amgen and Pfizer eliminated their neurology divisions completely. In the case of Amgen, I was able to talk with not the CEO, but the person right below him. And the CEO said, I don't want to wait 10 years for the drug. And it takes too long to develop neurological drugs.
I have 2 questions. The first one is, how much is research being shared around the United States and around the world on aging and Alzheimer's, etc.? The second question is, what is your opinion of David Sinclair's recommendation about taking NAD?
Okay, so David Sinclair is an amazing young man, because I think he's probably 50 now. He looks like he's 15. So that's a great advertisement for what he's saying. The nicotinamide chemistry is interesting. I'm not convinced that that is a solution. And you know, if you look at all of these treatments, whether it's vitamin D, omega-3, and saturated fatty acids, what you find is it's really hard to find out in human populations what something does. I like to give the example of, testing a compound. So let's take hydrogen cyanide. You take hydrogen cyanide, you give it to 5 people, they drop dead instantly. You know it's a toxin. For these other ones, the effects are very mild, and you have to have, thousands of people and, tens of years to figure it out. And if you look at something like coffee, coffee's good this month, it's not good next month. I think coffee, in the long run, looks like it might be good, but also humans are different. We're each different at one base out of 1,000 in our genomes, and those differences probably are important in how we respond. And so if you really want to do it, you need huge groups to find out who's responding and who's not responding. And so it makes it really, really difficult to actually find out if something is worthwhile or not.
And that's the reason for the huge cost of phase 2 and phase 3 clinical trials. You need so many humans.
If something worked immediately, we'd find out, right? So that's the easy part, but most of these effects are relatively small, and the question then is, if it's a 5% effect, if you look at the distribution of, who's helped, they're often overlapping. So there's no guarantee, okay, even if there's an overall 5% improvement in the population, you're going to be helped or not. So that's a frustrating part. The other part is that I teach a course in research integrity, and the mantra of that course is don't trust anyone. I was telling somebody, if your mother says she loves you, check it out. And there are so many examples where things just are wrong. Someone published a paper in 2005, and the title was "Why Most Research Findings Are False." So it's tough. It's very tough. But things that work well, those things work well. So when people found out that stomach ulcers were connected with the bacterium, you took the antibiotic, and your ulcer went away. Boom. Okay. But I don't think we've been that lucky so far. And the sad thing with Alzheimer's are these treatments that were based on a fiber that David didn't talk about, beta amyloid. These are the fibers that make extracellular deposits. And I think David can correct me, but my reading the literature is that those extracellular deposits have nothing to do with disease. And there are drugs that can remove them, okay? Monoclonal antibodies that can remove those effectively, no change in cognition. So as David emphasized, the bad actor is tau. And so, or alpha-synuclein Parkinson's. And so that's what you have to go after, which means that we're, hundreds of millions or billions of dollars of NIH money and company money that's basically gone down the drain following the beta amyloid plaque. I guess I got away from your question, but
Maybe I can take your first question, which had to do with international cooperation. I think it's very strong. Scientists tend to be generous in helping others, sharing materials, reviewing papers, which is a lot of work, peer review, and cooperating in large projects. So it's all a big plus.
And there's a new development that I think is significant, which is, in biology it's bioarchive, where in the old days you send your paper to a journal where it might sit for weeks or months or longer and be reviewed. At this point now, you can send in your paper to bioarchives and it's published online immediately for everyone to see. Hopefully it gets published somewhere with peer review, but I think it's a mark of how collaborative scientists are willing to share their results at very early stages.
Okay, we have a question here.
Yes, hi. Thank you for those good lectures. This is Dr. Rose Malley from the School of Medicine. You had a slide about diet, exercise, and sleep. I know for a fact that exercise helps, and that certainly not getting fat doesn't help. But what about diet specifically? More vegetables and fruits, that's what our mothers told us. But is there anything more specific? Is meat bad? Is fish bad? Or is it better to be vegan or vegetarian?
Yeah, the answer is it's complicated. One person I love is a glycobiologist at UC San Diego who gave a talk on sialic acid. Sialic acid are carbohydrates attached to proteins. It turns out the sialic acid in humans and chickens is the same. The sialic acid in beef, pork, and what's the other name? Lamb is different. And so, in fact, those are the red meats. And so it's very clear that the reaction, that red meat is actually, which I just had, is not good for us. It's inflammatory because of those molecular differences. So we are learning some of what the specifics are for different compounds. But I think the other advice is you have your dinner plate, half of that should be vegetables. Because, if you do studies on coffee, for example, it's like 495 say it's good, 401 say it's bad. If you look at fruits and vegetables, it's more like 95 to 1. And so there is, I think, saying that fruits and vegetables are magic, I think, is not an overestimation.
Yeah, what is your view on the pathologic process that actually makes these fibers implicated in causing disease?
That's a key question which has not been answered. The major risk factor in all these diseases is age. So at age 65, about 1 person in 11 experiences dementia. And age 95, it's like 8 out of 10. So age is the major risk factor, but there are other genetic risk factors like one protein, ApoE. One type of ApoE is a major risk factor. There are probably environmental risk factors too. Some scientists believe that viral infection can be a factor. In the case of Parkinson's, it's clear that there are environmental factors such as pesticides. You probably recall that Central Valley farm workers have experienced a lot more Parkinson's. But your genetics are a major factor, so look to your ancestors.
Are there any other questions? Yes.
Hi. I'm just a layperson, but I've been reading a lot about the regenerative aspects of the brain. It's more flexible than previously thought. If the fibroids are successfully removed or broken up, can the brain then regenerate, say, in a person younger who has Alzheimer's? Or once the fibers, it's just whatever state was at the worst state, there's no possibility.
We're going to have to see, but I think once neurons are dead, they're dead. I don't see, and they don't evolve.
There are regions in the brain where you have neurogenesis going on in the hippocampus and in the ventricles. And so there you have some possibility of neuroregeneration, but I think in general, as David said, it doesn't happen.
Would that be the executive functions, or would that be just the limbic system type of basic biological?
Well, the hippocampus is the home of sort of short-term memory, and I'm not familiar with that literature of really correlating if you can actually make differences in it. I think one of the things in all of us is that our hippocampus is shrinking, in spite of the fact that we can make new neurons, and it's correlated then with neurodegenerative disease.
Are there any other questions? If not, let's,
I have a question.
who has a question?
One question.
Oh,
I'm Eby Award from the department of our speakers today. So it is established that sugar is metabolically important, and yet there are recent publications about the damaging effect connected to Alzheimer of increased sugar uptake. How individually those things are different individually, and how will be the boundaries established?
so talking about the toxicity of sugars, glucose, for example, as an aldehyde is chemically reactive, and that's reacting with our proteins all the time. There are actually repair systems that have been found that will help take away the sugar aggregates from proteins. And sugars also can degrade into compounds that could cause cross-linking of molecules. So we're absolutely dependent on glucose, right? It's the heart of our metabolism, and yet it has a dark side. And that dark side is in fact these reactions that are dependent on the electrophilic nature of glucose. So, it's like oxygen we can't do without, very toxic. Glucose we can't do without, pretty toxic. Water we can't do without, makes things fall apart. So,
Any concluding comments for us, or have you said what you wanted to?
We're looking forward to joining your association.
Stephen Clarke, David Eisenberg, thank you very much.